The sequential evolution of Ophrydium versatile begins with the fundamental divergence of the Phylum Ciliophora from other eukaryotic lineages approximately 1.2 billion years ago during the Mesoproterozoic era. This initial phase of evolution was marked by the development of two defining characteristics, a complex infraciliature and nuclear dualism. The infraciliature, composed of basal bodies and associated microtubule systems, provided the structural framework for highly coordinated ciliary movement. Unlike the simpler flagellar systems of other protists, this allowed the ancestors of Ophrydium to develop sophisticated swimming and feeding currents. Simultaneously, the evolution of the micronucleus and macronucleus allowed these organisms to separate their germline genetic material from their metabolic control center, facilitating rapid protein synthesis and complex cellular responses to environmental stressors. These early ancestors were likely solitary, motile hunters that inhabited the microbial mats and open waters of ancient marine environments. The metabolic demands of such a complex cellular structure necessitated the development of efficient food vacuoles and a cytosome capable of ingesting a variety of bacterial prey. Over millions of years, the selective pressure to optimize energy acquisition led to the specialization of the oral region, where cilia became arranged in specific patterns to maximize the capture of particles from the surrounding water column. This period of early diversification laid the groundwork for the structural complexity that would later characterize the peritrichids. As the lineage transitioned into the Neoproterozoic and early Paleozoic eras, roughly 800 to 500 million years ago, a significant shift toward a sessile lifestyle occurred with the emergence of the Order Peritrichida. This evolutionary path was driven by the selective advantage of occupying a fixed position in the water column, which allowed for more efficient filter feeding in areas with consistent water flow. The primary morphological innovation during this period was the development of the scopula at the aberral pole of the cell. The scopula evolved as a specialized field of kinetosomes capable of secreting attachment structures. While some peritrich lineages evolved contractile stalks containing my1-mes to lift the cell body away from the substrate, the lineage leading to Ophrydium began to experiment with the secretion of an extracellular matrix. This period also saw the refinement of the oral ciliary apparatus into a counterclockwise winding peristomial disc, a hallmark of the peritrix that creates a powerful vortex to draw in bacteria and organic particles. The transition to sessility required a complete reorganization of the cell's internal geometry, as the once posterior end of the cell became the primary anchor point, while the anterior end became the specialized feeding hub. This polar specialization allowed for a more sedentary existence, reducing the energetic cost of constant locomotion and allowing the organism to invest more resources into growth and reproduction. During the middle of the Paleozoic era, approximately 400 to 300 million years ago, the family Ophrydiidae diverged from other sessile peritrichs by committing to a colonial existence based on a shared gelatinous housing. This transition involved a critical change in the cell cycle during binary fission. In the ancestral state, binary fission resulted in two daughter cells that would separate completely, with one often becoming a motile teletroch to find a new attachment site. In the Ophrydium lineage, mutations favored a lack of complete separation, where daughter cells remained embedded within the viscous mucopolysaccharides secreted by the parent cell's scopula. This led to the formation of the first true colonies. The evolutionary pressure for this change likely stemmed from the need for protection. A large, gelatinous mass is much more difficult for small invertebrate predators to consume than an isolated, exposed cell. This jelly-like matrix also created a buffered microenvironment, protecting the individual zooids from rapid fluctuations in water chemistry, temperature, or physical turbulence. The chemical composition of this matrix began to evolve from simple mucus into a complex network of cross-linked polysaccharides and proteins, providing both flexibility and structural integrity. As these colonies grew, the internal zooids had to adapt to living in close proximity to one another, leading to the development of shared physiological responses and a more integrated colonial identity. As the Mesozoic era progressed from 250 to 66 million years ago, the internal architecture of the Ophrydium versatile colony underwent further specialization to accommodate increasing colony size. As the gelatinous mass grew thicker, the individual zooids evolved an extremely elongated, spindle-shaped morphology. This elongation was necessary so that the aboral end could remain anchored deep within the supportive matrix, while the oral end, containing the feeding apparatus, could reach the surface of the jelly to access the surrounding water. To manage this extreme length, the zooids developed highly sophisticated My1-Mes, contractile protein fibers that run the length of the cell. These fibers allowed the zooids to retract instantly into the safety of the matrix upon sensing mechanical or chemical stimuli. This startle response became highly coordinated across the colony, ensuring that the delicate feeding structures were shielded from potential damage. During this era, the species also perfected the production of the teletroch, a specialized dispersal stage that allowed the organism to colonize new freshwater bodies. The teletroch stage represents a temporary return to the ancestral motile state, equipped with a posterior ring of cilia that allows it to swim away from the parent colony, locate a suitable substrate, and begin the secretion of a new matrix. This dual life cycle, sessile colonial and motile solitary, provided the flexibility needed to survive in changing aquatic landscapes.